Hello again, this is Dr. Dan Guerra from VerEvMed. Today we're going to take a little bit of a detour from what I normally do in VerEvMed lectures and in our studio and discuss something that is more general. So rather than taking a client's question or a perspective that is associated directly with a research paradigm, I'm going to go back a little bit and talk about something more general. Now, I have a reason for doing this, as I do for everything, of course. And the reason is that I want to be able to talk about programmed cell death, which is the topic of this mini lecture, in subsequent videos that I plan to be presenting in rapid fire between now and Christmas. So this is actually being taped on the 10th of November, 2017, and I hope to be able to do 4 or 5 more videos before Christmas comes. So in order to do that, I want to make sure that we have a background in apoptosis, that is programmed cell death, and hopefully I can refer back to this video and you'll take a look at it. So why don't we get started? All right, so like I normally do, or have been for at least some time, I start off with a picture that is from my collection of photographs that I've taken recently. This is when recently we've been to Canyonlands in the beautiful state of Utah in the dead summer. And in the summer, Canyonlands can be really, really hot. So I don't know if this picture makes you see how it makes you feel like it's really warm there, but it was really warm. It was over 100 degrees that day, Fahrenheit, of course. Anyway, we will get back to maybe discussing it a little bit later. I also thought this was particularly a good thing to present for programmed cell death because it's quite xeric there. And although a lot of things don't seem like they live as abundantly as they do, let's say, in some mesic environment like, I don't know, the peninsula off of the state of Washington or the Oak Hickory climax forests of northern Minnesota, believe me, there is a great deal of life even in the canyon lands, as you can see from some of the plants that are growing right there on the edge of the rock. It's much more stark contrast to life and death there, and that's why I chose this picture for this mini lecture. So let's get started. Again, I'm Dr. Dan Guerra. I am the VerEvMed Chief Science Officer, co-founder. And we can talk about what VerEvMed does a little bit later. But let's get into this. So I shall don my glasses. So what I want to talk about is a topic that relates to how cells die. So we all know that all cells eventually die. In fact, the tissues and the organism that harbors those cells all die. All living things die. There's irrefutable evidence in that regard. So, but the way things die is much more nuanced. And so what I'm going to talk about today primarily is one type of death, and it's called apoptosis. Now, it's not called apoptosis. And a quick look at the Wikipedia entry for this erroneously claims that that's how one pronounces it. The word means before excision or before drop or before cutting. So the Greek word for cut is ptosis, okay, or tome. And we actually get the root word actually comes down to us way back to Democritus or before him, who was an atomist, a pre-Socratic atomist philosopher. And tome means to cut. So atome means uncuttable. And that's how we get the word atom, right? The atom was considered the discrete smallest amount of matter or substance that the Greeks could sort of imagine. And so atome or atom is the smallest indivisible particle, right? Now we know, of course, that the atom can be divided into electrons and protons and neutrons, and then even particles of all of that. But that's where the word comes from. So apo means before, and tosis just means to cut, okay? So before cutting. That's what the word really means. And therefore, think about how you pronounce it. It's apoptosis. Apoptosis. You don't say the P again. It's a diphthong. Get over it, and please now always pronounce it correctly. All right, so you have a cell sitting there, any given cell, and it has a nucleus, and it has mitochondria, and it has endoplasmic reticulum, and it has Golgi apparatus, a regular eukaryotic, let's say, mammalian cell. There is some pro-apoptotic trigger. Now, we're going to talk about what those triggers can be, but something happens to the cell. And in this particular diagram, I want you to notice that what happens right off the bat is that there's a potassium channel that's opened up, and potassium starts leaving the cell, and so does chloride ion, okay? So you have cationic and anionic movement out of the cell. Now, what that causes to deal with the electrostatic imbalance of losing all of those ions is that water rushes out of the cell, okay? So what you get then is the cell starts to shrink, okay, from its normal size here. Isodiametric means it's pretty much in each direction, okay, in 3 dimensions. It has similar dimensionality. It starts shrinking, and it starts shrinking and shriveling up in contorted ways. And so what eventually happens is all of the organelles, including the nucleus, start to get scrunched together. And as water leaves more and more, what happens is that the DNA starts to fragment. The DNA inside the nucleus starts to fragment. Eventually, this cell starts pinching itself off into what are known as apoptotic bodies. And these apoptotic bodies, or now subcellular regions of what used to be that cell, are now engulfed by circulating macrophage monocyte populations and phagocytosis, so that the cell is now digested and removed entirely. Okay, now you can have resident macrophages, of course, doing the same business. Now, why is that significant? It's significant because, note, the cell has not leaked its contents. Not leaking its contents is a heroic way of dying, because if you leak your contents, you're going to leak out a lot of proteins and anionic lipids and even some proteoglycans and glycoproteins, and all of those things can be antigenic. You have something antigenic, what's that going to do? It's going to induce a major immune response, and now you're going to get an inflammatory response, or could, which means the whole organism starts mobilizing and marshaling towards where that cell is dying, right? Apoptosis is more heroic. It says, okay, I'm dying, and I'm going to make no qualms about it. I'm not going to signal this is happening, except at the very local level, and the cell gets digested before anything really is noticed. Okay, so that's the difference. So some people call it suicide. Okay. So let's talk about the difference between apoptosis and necrosis. So necrosis is what some would call a pathological cell death. Dying cells actually don't contract, they swell, and then with the swelling them causes them to lyse or split open, and then the cellular contents leak out, and the result is an inflammatory response, as just suggested. Apoptosis, or physiological, or PCD, that is programmed cell death, as dying cells shrink, as I just showed you. They become engulfed by leukocytes, that I showed you a macrophage. They leave no trace and do not result in a pro-inflammatory response. So that's, generally speaking, the difference. So how do you get cells to die? Well, there's 2 major ways, okay? And you can always then dissect 2 more and 2 from that and 2 from that and make subsets, right? But let's talk about the very cardinal level. There's cell death by injury, like mechanical damage, like if you get tissue ripping open, or you get a bruise or something like that, some kind of contusion or some kind of blunt trauma to tissue that will cause injury. That's one thing. Then, of course, is the exposure to toxic or destructive biochemicals. Okay. Now, that's opposed to cell death by suicide. So it's injury versus suicide. Okay. And that's, again, arguably what they call, what the cell biologists want you to imagine that apoptosis is. Now, the suicide can occur one of 2 ways. It can be intrinsic or extrinsic pathways. Okay. Now, apoptosis, or programmed cell death, is carefully coordinated collapse of the cell. You get protein degradation, DNA fragmentation within the nucleus, followed by a rapid engulfment by the neighboring cells, as we talked about. Essential part of life, or apoptosis, that is an essential part of life for every multicellular organism, simple invertebrates to humans. In fact, even in fungi, okay? So not even, way below the animal kingdom, okay? So apoptosis plays a major role from embryonic development, okay, in utero, all the way to senescence or old age, when eventually the cells start dividing and the organism gets close and finally dies, okay? So apoptosis plays a role throughout the lifespan of men. Okay. So why should cells commit suicide? First of all, it's needed for proper development, as I've alluded to. Examples are the resorption of the tadpole tail. Okay, so remember that you have the tadpole loses its tail, it's completely resorbed. That is an apoptotic event. Likewise, in the human, for example, the formation of the individualization or interdigitation of fingers and toes in the fetus in utero during development, during pregnancy. That is an apoptotic event. Likewise, the sloughing off of the inner lining of the uterus in females is an apoptotic event. And the formation, which is always happening, of connections and synaptic clefts between neurons in the brain, that is also an apoptotic event. So we call that pruning or tailoring of the neural system that involves apoptosis. Okay, a specific localized cell death so that new connections and new networking may occur. Okay. So besides that, which is normal physiology, right? Apoptosis is needed to destroy cells, like the first example we looked at. So cells infected with virus or intracellular bacteria, rather than to leak out or to lose those virus to the extracellular compartment, cells that are smart and heroic will die by apoptosis. Likewise, there are intracellular bacteria. Those cells do not want to release that to the extracellular milieu, so it might be useful just to let those cells commit what we're now calling dogmatically somewhat suicide. So cells of the immune system also have to turn over. That means they have to kill themselves, and apoptosis is a perfect way of doing that. Cells with DNA damage. So cells get DNA damage just by aging or by DNA damaging agents, sometimes acutely. But that chronic or acute damage to DNA, if repair not keep up with DNA damage, the cells need to go away. They need to go away because if they start dividing, they can result in mutated cells dividing. And of course, we know that can lead to oncogenesis, tumorigenesis, right? Cancer. And that's the last thing we talk about. Beyond just DNA damage, where you might get the inappropriate milieu of proteins being expressed, cancer cells also, if they are noble, will kill themselves, right? But chances are they're not going to be. That's why they're cancer, right? So in order to understand this in a little bit more detail, we need to talk about the eukaryotic cell cycle and its control. Now, when I give this a normal biochemistry lecture, I talk a lot more about what I'm going to do today. I'm just going to give you a very brief overview. And if you've seen this before, which many of you probably have, all this is going to be just a blush with what you recall from either, early days in undergraduate or maybe one day in class in medical school or in graduate school. So there are 4 phases of the eukaryotic cell cycle. And there are nuances about that, but 4 major ones. M, mitosis, G1, which is the first gap phase, S, which means DNA synthesis, the S for synthesis, and G2 is the second gap phase. So what's going on here? You have regulatory cyclin proteins and catalytic, that is enzymatically active, cyclin-dependent kinases, or CDKs. And they regulate, actually, that cell cycle progression. And during that process, you get protein phosphorylation and dephosphorylation. Remember, covalent modification via phosphate. And you also get protein turnover or degradation. All of that is critical to the movement through the cell cycle. Okay, remember, cell cycle is ultimately going to lead in cell division in a normal somatic cell. You also need, of course, at the same time all this is going down, chromosome replication. You need DNA synthesis. That's what the S is for, right? But you need to replicate that genome with super high fidelity. no mutations, no errors, no gaps, that kind of thing. Try to keep also down the number of copy number variations also. Splice variation, all that stuff's got to be kept at a minimum. So you get chromosome replication and segregation. And of course, that's fundamental to all eukaryotes, okay, organisms with a nucleus, from yeast to man. And all of that is controlled by these heterodimeric protein kinases, these cyclin-dependent kinases, or CDKs. So here's a nice cartoon of how that's going on. Let's take a look at the beginning here, where you have the G1 phase, where you have centrioles replicating. Ultimately, you end up going into an S phase, where you start synthesizing DNA. Then you go to a final growth and activity before you get cell division. And then you get a series of steps leading to mitosis, from prophase all the way to cytokinesis, where you have a new cell born, right? So this is a very, very, very truncated version of what's going on in the cell cycle. So I want you to keep that in mind, because the cell cycle during that cell cycle, there are checkpoints. And during those checkpoints is where a cell is going to continue to go through that cycle and end up dividing, or it's going to arrest and it's not going to go any further. Now, the apoptotic pathway is going to be embedded within that, okay? All right. Now, much of what I'm going to talk about now for the rest of the talk is going to come from the annual review of pathological mechanisms of disease. It's a paper it's a review. It came out 7 years ago, but this is a lecture, just a review of programmed cell death, right? I'm not going to talk about all the hot topics. That's what I'm going to do subsequent from now until Christmas. So senescence, okay, when cells age, is a tumor suppressive mechanism that permanently arrests cells at risk for malignant transformations. So senescence is where a portion of that cell cycle is arrested. It's blocked, stopped. However, current evidence shows that senescent cells can nevertheless have a pathological consequence within a microenvironment. Most significant of that effect is an acquisition of what's known as SASP. Love that word, or I love that acronym, SASP, senescence associated secretory phenotype. What that does, that's a cell's phenotype. That means it's a way the cell expresses itself during the senescent process. It can turn senescent cells like fibroblasts into pro-inflammatory cells. Yeah. And those cells have the ability to promote tumor progression, because they're going to be sending out signals that are not the kind of signals you want when a cell starts to senesce. So the SASP process can alter the normal senescent process and signal far distances from one cell mass to another, and ultimately lead to a progression of cell cycle, and maybe, if there are mutations in those senescing cells, to outright tumorigenesis. Okay, so SASP is something that's been looked at quite a bit. So let's take a look at this. This is from this annual review's paper. So you get an intrinsic or extrinsic genotoxic stress. Okay, so that can be things like heavy metals, or UV radiation, or ethanol, right? Or some other kind of cytotoxic agent, right? And what that does is that's going to damage, what are known as sensors and transducers. That means basically receptors, and then all the signal transduction cascade that comes from the receptors associated with membranes, plasma membrane, and all the intracellular endomembranous systems. Ultimately, very first step, you're going to trigger the expression activity of a protein called P53. Okay, P53 is a tumor suppressor gene. Now look what it does. It's going to block the SASP phenomenon. Okay, and rather than do that, it's going to go into senescence arrest. Okay, what that's going to do is block any possible cancer. Okay. Likewise, before it even gets to senescence arrest, it's going to go ahead and cause a transient arrest and maybe repair. So what P53 tries to do is give you a little bit of time and give the cell a chance to recover. If that cell can recover by DNA repair, which is usually one of the major because it's a genotoxic signal, which means the nucleus is recognizing there's something happening to the cell, and the first thing it tries to protect is the genome. Right, so usually that involves some associated damage to the DNA. So repair mechanisms are turned on. So that's what you want to have happen. When that happens, you get no cancer. Now, that's the best of all worlds. However, if SASP is turned on, if P53 is not able to dominate this whole process, SASP is turned on, and there's two aspects of it: early and late. Early, you get alarm signals in tissue repair, and that again is a stop. What's going on to the cell? What's going on in this tissue? Let's stop. Let's repair what we've got, and let's remain senescent. Okay. And that will, of course, block cancer and probably also the continued aging of that and any pathologies associated with it. But later on, you get what's called chronic SASP, and that then gives you a persistent inflammation, which is going to flip this whole thing completely on its back. And now we're going to get cancer. We're going to get cancer promotion. We're going to get aging promotion. We're going to get all kinds of pathologies. Okay. So what does it say down here? DNA damage signaling pathway leads to the activation of the P53 tumor suppressor. Activated P53 triggers cell fate decisions such as senescence or apoptosis, right? Depending on the context, that is what else is going on in the cell and the cells associated with the cell that's being signaled, you're going to get P53 restraining senescence associated SASP, like we just said. And regulation of the SASP by P53 suggests a cell non-autonomous function of that tumor suppressor. In the short term, the SASP may promote tissue repair. In the long term, it can cause chronic inflammation, and it can actually trigger then cell division. If those cells are mutated again, you're going to get cancer. So here's a much more kind cell cycle diagram. Kind meaning it's more accurate. Okay, so I consider things more accurate to be kinder than when things are dumbed down. Okay, just the way I think. That's how I think as a scientist. So first of all, you get a checkpoint here. Now all of these red colored or whatever they are, I guess they're red colored pathways. They inhibit cell cycle progression. So anything that can cause an associated blockage of cell cycle progression is going to block at various points in the cell cycle, right? So you have what I call checkpoints. So here's one checkpoint here, right at the entry of the M phase. And the DNA damage checkpoint's going to happen right here. And all of these are individual proteins that are turned on. ATM turns on P53, turns on P21. That all is going to block cyclin from progressing through the cell cycle. Down here at this intra-S phase checkpoint, okay, you're going to get ATR turning on check 1, blocking CDC25. When that's blocked, you're not going to continue to enter into it. You see how this works? Spindle assembly checkpoint with the MAD protein that's going to block this whole process of polyubiquitinylation of a protein called securin, so you're not going to move through anaphase. Spindle checkpoint here in position number 3, okay, it's going to block CDC14, so you're not going to get to that same system. You're not going to get through telophase, and on. There's P53 again, and P53 down here. Multiple checkpoints, okay? Any place in those checkpoints, as I'm saying, that's a possibility where the cell is now going to marshal in new phenomena, and the new phenomena can be programmed cell death. Okay. So that's the important thing. So I wanted to take you down. I wanted to get that, I wanted to take the lens and I wanted to give it high magnification and show you what's going on at the molecular level. Now that's a very brief overview, right? It's a very simplistic overview, although it was kinder because it was more accurate, as I just said. I can do a lot more in there. In fact, probably spend a good hour just discussing how cell cycle checkpoints work, but we're going to do that some other time. And again, I do that in general type of biochem lecture. Let's go back to talking now. Let's pull that lens back out, give you a larger lens and look at the whole picture. Again, I want to reemphasize that apoptosis is different than necrosis. Necrosis is swelling, membranes break, ATP gets depleted. Now, if you've been following along, that means AMP kinase can be turned on. Following along meaning in previous talks I've given that are all on video in case you want to see this. When AMP kinase is turned on, remember you can get into an autophagy. A cell can start ingesting parts of its cytoplasm, yet that's not apoptosis. It's not ready to die. It's actually it's taking what is existing in the cell, removing it by digesting it by proteases and glucosidases and lipases, tearing down all the macromolecular structures. But at the same time, it's allowing for the expression of other genes that are going to allow that cell to survive. And that's all a biofuel economic situation, as well as dealing with overall abundant cellular stress. Now, again, that's quite different than apoptosis. Here you get the cell condensation, you get, remember, the water leaking, the shrinking. This whole process actually requires ATP. So in order for apoptosis to work, you need to burn energy. So the cell has to be very, very controlling here, right? It has to go through this cascade of events that says, I'm going to use up all the ATP I've got, and there's no coming back from this. There's going to be a point reached where the cell is just going to commit suicide. It's going to die, and you can't get back from it. That's basically what happens. So here's kind of another way of looking at it. So you get cell injury, and that can include disease, right? A pathology, or maybe an oncogenic effect, or maybe an autoimmune response. So cell stress can be looked at from two different valences. One is dose intensity, or the acuteness of it. And the other, of course, is cell vulnerability. It's not just acute. That also means if it's a chronic, constant, right, stress. And cell vulnerability. Some cells are far less vulnerable to stress than others because they have a lot more control over how they receive those stress signals. Anyway, the two things can happen. You can get injury. If there's enough injury, we just went through this. The cell cycle is going to make a decision somewhere where there's a point of no return, kind of like Plato's divided line, right? And you go from reversible, like you can go back and stay and you can maintain a cell, like what autophagic responses can do. Or it's irreversible. Goodbye. We're going down this pathway and we're going to commit cell death, okay? Programmed cell death, apoptosis. There's also an adaptive possibility, and that's going to be metabolic and structural. That's going to allow for this injury pathway to proceed, but never going to hopefully get to the irreversible domain, right? Now, I told you, and I've been trying to allude to the fact that apoptosis is somewhat complicated, okay? And apoptosis is known as having an extrinsic pathway, intrinsic pathway, and then there's this other whole system which basically involves etiologic agents, that is pathogens. So let's take a look at the extrinsic pathway first. The extrinsic pathway works through a plasma membrane-bound protein complex called death ligand and death receptor. So a ligand, polypeptide, binds to this receptor, and then quickly adapter molecules are associated with this receptor. Disc formation occurs inside the cell. A protein called caspase 8 is activated. It activates caspase 3. Okay, so these are proteases. Caspase 3 is known as the execution pathway. What that does is endonucleases are activated. Remember, it starts chopping up the DNA, generating what looks like a ladder on a agarose gel. You get the degradation then of chromosomal DNA. You also get protease activation. So you got endonuclease breaking up DNA, protease breaking up protein. You get a degradation of nuclear and cytoskeletal proteins. That causes cytoskeletal reorganization. This is leading down that pathway. You start seeing cytomorphological changes. So you can see this under a microscopic examination. You get chromatin and cytoplasmic condensation, nuclear fragmentation. I know this sounds familiar by now. Ultimately, you get the formation of apoptotic bodies, and goodbye. That's it, right? Now, that's the extrinsic pathway. That's like the quick way. It's like, oh my gosh, we got a death ligand bound to the death receptor. Quickly, let's lead to caspase 3 activation, die. The intrinsic pathway is slower. That can include things like radiation, okay, such as ionizing radiation, even UV radiation, various kinds of toxins, right, biochemicals that cause toxicity to the cell, and also hypoxia, which just means removal of oxygen. Imagine this like if a person becomes unconscious and you start to get a lack of oxygen going to the brain, okay? This is the kind of thing that happens, right? So as long as those cells are still alive and they can carry out apoptosis. So what happens in the intrinsic pathway is, I think, far more interesting because it's more nuanced and it involves multiple more proteins. But there are changes directly to the mitochondria. Now, the mitochondria, remember, that's the subcellular organelle that's involved in the TCA cycle, electron transport chain, oxidative phosphorylation. It makes ATP. But the mitochondria does a lot of other things too, okay? It sort of controls amino acid metabolism. It controls a lot of lipid metabolism. So the mitochondria isn't just, that terrible term that textbooks use called the powerhouse of the cell. I wasn't going to use that. I did just to placate a particular person I know. But the mitochondria does much more than that. And when the mitochondria starts receiving signals that something's going on outside, right? And that outside mechanism is going to start telling the cell, get ready to become apoptotic. Once that mitochondria starts going through a degradatory pathway, that is going to lead ultimately, again, through a series of steps beyond which you cannot get out of, and you're going to kill yourself. The cell's going to go through apoptosis. Notice it goes through a caspase-9 activation, but then after that, once you get to the execution pathway, same thing happens. Now, third thing that can happen is you can get an invasion, a pathogenic invasion. And if that happens and T cells are recruited to the site, you can get cytotoxic T cells. This is a particular subclass of T cells, which are part of the adaptive immune response. These cytotoxic T cells are going to have a couple of interesting proteins called perforin and granzyme. I like that's like grandma's enzyme, right? Anyway, the perforin is going to poke a hole into the membrane. It's called perforin. That's why it's called that. And it's going to release granzymes B and A. Okay, so this whole cytotoxic T cell is going to bite into the plasma membrane of that cell. It's going to open up a hatch or a pathway. It's going to send in these two enzymes. Enzyme B is going to activate caspase 10, and both granzyme B directly and caspase 10 are going to turn on the execution pathway. Granzyme A is going to go through another thing called the SET complex, and that's going to take the business of cleaving up the DNA. So same thing here. What you've got is an intoxicated cell. That's why the T cell was recruited. Okay, you have an adaptive immune response. The cytotoxic T cell is going and saying, okay, let's take care of business. We've got to kill this cell. Okay, and so it does it by opening it up, introducing granzyme, grandma's enzyme, and carrying out the process, ultimately leading to cell death and programmed cell death at that. Here's another more colorful, a nicer picture of the same thing that's going on. Now, this came from a recently published paper. In fact, it's available online right now. It comes from the journal called Pharmacology and Therapeutics. This was available just a few days ago. I'm recording this on the 10th of November. This became available on the 8th of November, 2017. So here's your death ligand system here. You notice they got the geneticists had some fun. The RIP, right? Rest in peace protein, right? And you've got the death signaling, right? That's what this is supposed to be, death and destruction signaling. You've got this thing called FASL, which is the ligand that binds to this death ligand. Here's your extrinsic pathway working here. This is extrinsic. Remember, it goes right to caspase 3. No fooling around. The cell kills itself. The intrinsic pathway, I told you, much more nuanced, much more flavorful, colorful, as the slide shows. It's working in the mitochondria, right? The intrinsic pathway. I told you the mitochondria is very complicated. Look at all the different proteins that get fired up here. Some of them have to block the anti-apoptotic signaling, which is what this BCL-XL is here, blocking that, because the mitochondria is always poised to start to say to the cell, we are going to go through apoptosis, because if it's not poised, then there's no way a signal can be transduced in any kind of timely temporal manner. And remember, this has got to happen fast if you want this cell to kill itself, right? So anyways, it stops the normal maintenance of the mitochondria, turns on this pathway here. This pathway ultimately leads to, rapid movement to accumulation of all these proteins. There's a Diablo protein, another one. I know the geneticists are having a little bit of fun with this, right? They block certain pathways that would naturally block the apoptotic transgression. Blocking that allows caspase 9 and caspase 3 to work, and there you get apoptosis. There's the movement. There's the exocytosis, or that is the removal of cytochrome C from the mitochondrial membrane. Once that happens, that protein free in the cytoplasm triggers the APAF caspase-9 response. It's all part of the intrinsic pathway. DNA damage directly can lead to that, can turn on this system here. When you turn on that system, you short circuit these more standard external sources and move right through this pathway, building up these proteins that ultimately are going to block something that would normally block apoptosis. You open up apoptosis and you die. Okay, so this is some of the more detail I was promising you. All right, now, see, I told you that was going to be a quick romp through apoptosis. Hopefully it wasn't too fast, and hopefully I didn't gloss over the things that you may have wanted more information on. Ultimately, as I said, I'm going to open up the apoptotic pathway in papers I'm going to discuss that are going to be front and center, recently published, that are going to deal with specific discussions of oncogenesis in particular, is what I plan on talking about up to Christmas. This is, again, our trip to the Canyonlands. You can see this rail protecting the author from not falling over and probably having some kind of programmed cell death as well, or tissue damage for sure. Remember that VerEvMed, okay, is scientists verifying published evidence in medical biosciences. So that's what we do, okay? So all of these lectures that I give are just giving you a demonstration of the kind of thing we bring to the table when we deal with your individual questions about what this current research says. And we look at the evidence and we vet that evidence, we verify that evidence, we put together a summary of what that evidence says in the current published literature for given questions about biomed, pharmaceuticals, and other large aspect of topics in biology, biochemistry, medicine. So thanks much for your attention. And I'm going to close here and say thank you very much again. And my current sign off is bye for now.